Hi everyone. Today we're going to look at cell division and replication. I'm just going to quickly check in with this lighting, which looks good. Okay, so cell division and replication, some of this is going to be a bit of a review from last year, particularly when we look at mitosis, but we're going to do a quick recap on the types of cells that exist in a sexually reproducing adult organism. So let's have a look at this first. Each species has a particular set of chromosomes and a particular set number of chromosomes. So the number of chromosomes and the type of chromosomes and the way that they're arranged and ordered is very much a key indicator, or something that we use to identify different species. So for example, all humans will have 23 chromosomes. Well, in general, the vast majority of humans will have 23 chromosomes, 23 sets of chromosomes, and 46 chromosomes in total. So sets, pairs of chromosomes, that's going to become a bit of a key feature when we look at our mitosis and meiosis. So 23 pairs of chromosomes, 46 chromosomes in total. So sexually reproducing adult organisms have two types of cells in their body. They have both diploid cells and haploid cells. Diploid cells, we're going to have a quick look at those first. These guys here are body cells, and the body cells, I'll take this all the way down here. Diploid cells are body cells. They have pairs of chromosomes inside of them. So 2 chromosomes for the same trait, and that's why we call it a pair. This pair and these 2 chromosomes, one of them comes from mum, while the other one comes from dad. Each have their own information about the trait. For example, mum's chromosome might provide information about blonde hair. Dad's chromosome might provide information about brown hair. Both of these chromosomes refer to hair color, but the DNA of the genetic information that is actually given on them can differ, but they're both about the same trait. These chromosomes, because they're both information about the same trait, are called homologous chromosomes. Homo meaning same, so it's an easy way to remember that. Same types of chromosomes. So diploid cells, 23 pairs of chromosomes, 23, that means that 23 chromosomes are from mum, 23 chromosomes are from dad, and each one of them has a partner. In contrast, haploid cells are sex cells or gametes. So what that means is these are the egg cells that are found in females and the sperm cells that are found in males. Key difference between haploid and diploid cells is that haploid cells have half the amount of chromosomes that are found in diploid cells. This means that while there are 46 individual chromosomes or 23 pairs in diploid cells, haploid cells have 23 individual chromosomes, so they don't have any paired chromosomes in them. This is particularly important because of the purpose or the role that gametes play in reproduction. So, in sexually reproducing organisms, an egg and a sperm have to fuse to create a zygote in order for offspring to be produced. If the egg and the sperm have half the chromosomes, 23 chromosomes and 23 chromosomes, so that when they do fuse together, they end up with a total of 46 chromosomes, which is exactly the total that we need for all of our diploid cells over here. If the egg and sperm were to be diploid cells, when they fuse, they're going to have way too many chromosomes. All right, so super important that these guys here have half the number of chromosomes in order to make sure that they end up, the result is that they end up with the 46 chromosomes. Let's have a look at this. So what we're going to be looking at now is how each of these types of cells replicate or are produced. So what we'll do is we'll have a quick recap on this side over here when we look at our body cells with the 46 chromosomes, 23 pairs, and how they're produced, and then we'll come over across and have a look at meiosis, which is our new topic for this year. So let's have a look. Mitosis. Mitosis and meiosis both begin with the same step, and in fact it's actually not considered to be part of either of them. It's something that cells exist and do in between phases of mitosis or going through meiosis. So interphase is is the main phase in which cells exist in. In fact, they spend 90% of their life in this phase. During this time, they are producing protein, they are repairing, they're replicating DNA, and they're replicating the number of organelles that they have. So when we're replicating DNA and replicating the number of organelles, it usually means that we're getting ready to divide the cell. In the process of replication here, I've written down that it replicates the DNA, but it doesn't change the number of chromosomes. What that means is, and you're going to have to forgive me for using two different colored blues, is that if I have my DNA strand that's here, and the DNA strand, we know that when it's unwound and it's not tightly coiled up, that DNA strand is just a long strand of DNA. If that DNA strand is to wind up and condense and condense and condense and condense, that DNA strand before replication will look like that. It might look a little bit wriggly, but it'll be one line or one tube of DNA. That's one chromosome. If the DNA strand has gone through replication, where it has unwound and unzipped, and it's been through the process of creating two strands of two pieces of DNA that are identical, what they don't do is unwind and unzip and create two completely separate pieces of DNA. So it doesn't actually separate. This DNA, however, will unwind and unzip, and as it begins to replicate and become two, it will actually stay connected at some point in the DNA. And when we have a look at them, when we see these chromosomes, these replicated chromosomes or replicated DNA actually look like X's. So when the DNA replicates, yes, it replicates, but they actually stay connected. So therefore, the number of chromosomes stay the same. So instead of having 46 of these sitting inside of them, they're going to have 46 of these. Still one chromosome. Okay, so after it finishes with prophase over here, it heads into interphase. It heads into prophase. So during prophase, the nuclear membrane breaks down, the chromosomes condense, and they find their homologous pairs. So you can see the difference here between the DNA that's all just sort of a bit of a mess inside the nucleus. It's been through and it has replicated. In prophase, we've got it here, they're condensing and condensing and condensing and finding their homologous pairs. So the homologous pairs that we might have would exist. Remember, homologous, mum's chromosome and dad's chromosome. So these two might be the mum and dad homologous pairs of chromosomes. So condensing, finding their pairs. During metaphase, which is our next phase, these chromosomes will then line up in the center of our cell. So they will line up in the center of our cell during metaphase. So meta, middle, nice and easy to remember. The spindles will then attach from each side of the cell, and they'll attach to each one of the chromatids. So these individual bits are called chromatids. Then during anaphase, which is our next phase, those spindles will pull, and they will pull apart the chromatids to each side of the cell. After that, we enter the very final phases of telophase and cytokinesis. Telophase meaning two, so we end up with two separate membranes, and cytokinesis meaning we end up with two separate cells. Now, pretending that this is the same blue and that this is the same red, you should be able to see that we have now got two identical cells, one with a red one with a blue one with a red one, and one with a blue one. And so we've gone from one parent cell into two daughter cells, and the daughter cells are identical with the same number of chromosomes. So that is our mitosis process. Mitosis is typically used for where we need this more of the same type of cell. So this would be growth. So when your bones are growing, we need to produce more bone cells. And so therefore the bone cells that exist there will then replicate and divide and split, and so that there are more bone cells to help you and allow for you to grow. And the other situation is going to be repair. So if you cut your knee or graze your knee and you need more skin cells there, your skin cells will then divide and split in order to create more identical skin cells. So that there is diploid cells, body cells being reproduced by mitosis. Let's have a look at the new stuff now. So haploid cells, we know that they have half the number of chromosomes. So these guys here are produced by meiosis, and they go through very similar phases to mitosis, but they go through two sets. So over here, you should notice that interphase has been written on both of them because these cells are going through interphase 90% of their life. And then you should actually notice that we have prophase 1, metaphase 1, anaphase 1, and telophase and cytokinesis. So telophase 1 and cytokinesis. This side here is actually called meiosis 1. Then it goes on to prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis. And this side here is actually meiosis 2. So you can see very, very similar processes. We just have 2 rounds of it. And there are another couple of key features. So you're going to have to pay pretty good attention in this section here. So let's start with our interphase. So before our interphase begins, we have our chromosomes, and this all begins with a diploid cell. So before we end up with a haploid cell, which is what's produced at the end, at the beginning we have diploid cells. So these guys here, they have 46 chromosomes, 23 pairs, 23 from mum and 23 from dad. So I'm going to model this with just two chromosomes. So in interphase, we know that DNA is replicated. So during the process of DNA replication, our chromosomes go from looking like this to looking like this. Okay, so a little bit rough, to looking like that. So we have our diploid cell at the very beginning of meiosis that has replicated DNA, 46 chromosomes, all looking like this. Then prophase 1 begins and the nuclear membrane breaks down and the homologous pairs align. So if these two here are the exact same chromosomes, so homologous pairs, one mum, one dad, it's about the same type of DNA, they're going to pair up. Then what they're going to do is they're actually going to get really close with each other. And during this process, they're actually going to not collide so much, but actually get super tightly connected. And when this happens, the DNA from these chromosomes will actually swap. And they will swap in order to increase variation. So this process is actually called crossing over, and the process of the DNA swapping when those chromosomes are actually meeting ensures that you, for example, do not look identical to any of your brothers or sisters. Without the DNA crossing over part, without this crossing over process, every time meiosis would go through, the exact same chromosomes would be being split off. And so basically you'd end up with 4 variations of that particular trait, and that's all. So this process of crossing over is really, really important to ensuring variation amongst the species. And it's actually one of the benefits and advantages of sexually reproducing organisms. So we'll move on. The chromosomes, they get really, really close to each other, and they end up crossing and swapping some of their genetic information. After this point in time, the homologous pairs move into metaphase, where they align up in the middle with their pairs. So these guys here, we've already said they are homologous pairs. They've been through a process of crossing over, and I'm sure you can see that DNA has actually been swapped between them. They're no longer the same color. Once they're in their pairs, during metaphase, the spindles reach out and attach to them. We then go into anaphase where it pulls them apart and they move away. So anaphase, away. They move away from each other and pull themselves into two separate cells, or they begin to form two separate cells. So from anaphase, we move into telophase and cytokinesis. Telophase meaning two, and cytokinesis is where our cytoplasm begins to create its own cell. So after meiosis 1, we now have two cells, two daughter cells. These daughter cells no longer have 23 pairs of chromosomes. They are down to 23 chromosomes. They are also not identical to each other. So we've now hit our second major difference to mitosis, the first one being this process of crossing over occurring. We're going to head into prophase 2 now, where again, it's very similar to mitosis. They're going to condense and they'll be visible as they are now. The chromosomes will then line up in the middle. So you can imagine that you would have 23 of these chromosomes all lining up in the middle. The spindles will then attach to each one of the chromatids. So remember, we talked about these guys being replications of each other. The spindles will then attach to each one of the chromatids in metaphase before anaphase occurs and the spindles pull the chromatids apart. So what that will look like for this one is the spindles will attach to the chromatids and pull them apart. The spindles will attach to these chromatids and pull them apart. Then we go through the process of telophase and cytokinesis, where this one here is now its own cell. This one here is now its own cell, and its own cell. So we can actually see now that we have created 4 daughter cells. All of them, none of them are identical, so they've all got variation. And we can also see that these guys here do not have the full set of chromosomes. So we're down to 23 individual chromosomes in each cell. I hope that was clear, and good luck with your understanding of the differences between meiosis and mitosis and the major outcomes. Key things, remember, meiosis has 2 phases, 2 runs, meiosis 1 and meiosis 2. It has a crossing over part in which increases variation, and it also produces 4 non-identical daughter cells as opposed to mitosis's 2 identical daughter cells. Good luck.